Hi everyone. I'm Michael Carter. I'm a senior principal computational chemist at Domainex. I've been at the company now for around a year, and I thought it'd be good to take you through some of the sort of projects we've been working on and how we've been applying our computational and AI ML platform to expedite the drug discovery process in these projects. So the first project I wanted to touch on is an allosteric discovery project. This is a project we're working on with a client, and it's a really hot area for drug discovery at the moment. There's lots of new patents coming out pretty much on a weekly basis. And obviously these patents are captured by Reaxys or SureChemble because they're very new. So how can we extract that data from those patents to help guide our medicinal chemistry efforts? One thing we can do is to utilize our proprietary AI ML algorithm to extract patent data. This uses a technique called optical structure character recognition for anybody who's interested, to take compound structures and convert them into machine-readable SMILES. We can also do this with data tables as well. And what this has enabled us to do is to really extract the wealth of new allosteric series patent data that's being published. And this has allowed our medicinal chemistry teams to interrogate the SAR and to hopefully begin designing novel allosteric compounds of our own. In addition to this, we've utilized the data from these patents to begin building QSAR models to understand if we can start predicting potency of allosteric inhibitors of this target. But it has opened up a lot of other questions, like where do these compounds bind? So we've also been looking at how can we apply our kind of computational platform to understand potential binding sites of these allosteric molecules. So we've looked at binding site detection. We tried conventional approaches, but they tend to struggle with these more cryptic or allosteric pockets. But we've implemented two different kind of approaches, one of which involves machine learning, so looking at scoring residues for their probability to be involved in a cryptic or allosteric pocket. The other involves molecular dynamics, so sampling the protein using physics-based approaches and understanding where druggable cavities lie. When we did this on the protein of interest, what we found is the machine learning and the MD-based pocket detection really lined up well. So if you look at the area that I've highlighted on the two images, you can see that the AI-ML model has a high probability of an allosteric pocket here, so lots of red coloring. And the MD pocket analysis, the red surface, is where it predicts a highly druggable allosteric pocket. In addition to looking at pocket detection, we can also look at applying protein ligand co-folding methods. So we also did this as well, and interestingly, the co-folding also picked out this area of the protein as a possible binding site for these allosteric molecules. And what we actually managed to do, literally, I think it was a week after we made these predictions, was solve the crystal structure. So the molecule that's shown in cyan is the actual crystal structure of this small molecule in the allosteric pocket. And if you compare the AI prediction to the X-ray, you can see we've actually got a really pretty good agreement with the ground truth. Obviously, it's not perfect. The RMSD is around 3 angstroms. But what this kind of represents is how we can begin to use these machine learning models and also our pocket detection to tease out where these compounds could be binding. But great to always have the experimental validation as well from our structural biologists. And it really just highlights the synergy between the computational predictions and how we're able to confirm those with our wet lab experiments as well. So now we've kind of got the data, we understand where these compounds bind, we're beginning to use this information to really push for discovery of novel allosterics for the client we're working with on this target. So we've been able to run virtual screening by developing pharmacophore models, as well as using molecular docking, but also looking at techniques like scaffold hopping and QSAR modeling to guide the design and optimization of new allosteric compounds. And I think really, just to kind of highlight that the CAD platform was quite instrumental in identifying this, and it's really nice to see that these techniques are really being fruitful for our client projects. The second project is more of a lead optimization task, particularly focused in on ADMET optimization. So this was a project we worked with a long-standing client. And when I joined the project, it was in a late lead optimization stage. And there were several flags with compounds that were coming out of this project, particularly around metabolism, both CYP-mediated and human aldehyde oxidase-mediated. And there were several key physchem properties that needed to be maintained, such as log-D and CACO2. So we were asked to identify and deploy tailor-made machine learning models to hopefully begin to understand this SAR and to utilize them in compound design and optimization. So we actually implemented a whole host of kind of tools to understand metabolism. So we implemented several sites of metabolism tools and metabolite predictors, both CYP-mediated and human aldehyde oxidase-mediated. And these were able to identify sites of metabolism that were pretty accurate, but what they couldn't do is understand how distal effects far away from the site of metabolism actually could impact that site of metabolism. So what we actually had to do was look at other approaches to maybe enhance our capabilities. So we implemented a tool called ESNUL, or Estimation of Nuclear Philicity and Electrophilicity, which is a QM-AI model which tries to understand how happy an atom is to be protonated or deprotonated. And what we found was that we were able to actually understand how these distal kind of changes impacted the site of metabolism we were interested in, and reducing the ability of a specific nitrogen to be protonated actually increased our metabolic stability, but it also increased our on-target selectivity. And what these models really enabled us to do was interpret the SAR on this project and guide the next designs for MedChem as well. And as I mentioned previously, log-D was a really important descriptor. So having a log-D of between 1 and 2 was crucial to obtain cellular potency for these compounds. And the client had a lot of log-D data, and we tried to model it with commercial and open source models. And these are the sorts of results we saw. The models weren't great, the performance was poor, and we couldn't really use these models for compound optimization. So what we decided to do instead was to take the client's data, to utilize our QSAR platform we have, and to build our own models to model their local data. And what we managed to do was design models that were far more predictive for the client's data, and were able to be used in compound optimization. This project also recently nominated a successful clinical candidate, as well as some really promising backup compounds as well. And I think what this highlights is the ability to provide our clients with the best models for their project at the specific time. The next project I wanted to touch on was the RNA project, which Nick kindly has already introduced. So we ran a virtual screen against the SAM6 RNA using our internal proprietary fragment library that contains around 1,200 fragments. And we developed several models to understand how these fragments could potentially be binding to the SAM6 RNA. Broadly, we could split these into ligand-based and structure-based models. So we developed ligand-based models to understand the shape and electrostatic complementarity to known SAM6 binders. And we also developed pharmacophore models to understand key features of molecules present that could bind to the SAM6 RNA. And what these ligand-based models do is enable us to rapidly screen not just our fragment library, but hopefully in the future larger libraries against such RNAs to identify possible binders for these targets. In addition to this, we also implemented some structure-based workflows. So we have access to some Schrödinger software, including their docking tool Glide. And what this enables us to do is perform high precision not protein, RNA ligand docking, and to obviously use the scores that are generated for that as another method to rank how these compounds could interact with RNA. We also integrated a machine learning model, which is a deep learning algorithm called RNA Amigos 2, and this is trained on RNA ligand binding data to take in a 3D RNA binding site and predict how the probability that that compound could bind to that binding site. So we're kind of trying to combine our physics-based and AI/ML-based derived interaction likelihoods to enhance the prediction of our structure-based drug design workflows. And Nick's already stole my glory on this, but we got a really nice hit rate of around 30%. And this was leveraging all of the different predictors we'd put together, so our ligand-based scorers, our structure-based scorers. And I think what it highlights is the kind of the suite of computational and biophysics tools we have can enable discovery against these kind of novel RNA targets. So yeah, really nice use case. And lastly, I think almost every talk has mentioned PROTACs and molecular glues. Maybe Jason didn't. But PROTACs and molecular glues are not in your traditional kind of rule of 5 chemistry space. So we need to understand or develop novel descriptors to understand these types of molecules. So we've looked at both ligand-based and structure-based descriptors and models for these types of modalities. And one of the kind of ligand-based metrics we've introduced is called SMID, or smallest maximum interaction distance. And this is a measure of how compact a compound can be in three-dimensional space. A lot of people call this chameleonicity when it comes to PROTACs and molecular glues. But this can be quite favorable in terms of shielding polarity and making your PROTAC or glue more permeable whilst not sacrificing solubility. We've also implemented a machine learning approach for 3D polar surface area prediction. And the plot on this slide is showing a performance against a very challenging external data set from Novartis. And you can see, hopefully, Spearman's row is quite small on my screen at least, but it's around 0.96. So a really, really strong positive correlation between our predictions and EPSA, which is really nice to see. And I think AJ touched on this in his slides, where we can start to use these metrics in combination with other phys-chem properties, such as log-D, heavy atom count, other sort of phys-chem properties, to create really powerful composite metrics to guide ligand-based optimization of these PROTACs and molecular glues, particularly focused around permeability, solubility, and efflux. And in addition to this, we've also started to look at ternary complex prediction. Now, this is very challenging. Anyone who's tried it in this room will know. The protein folding method I mentioned earlier for protein ligand folding cannot do this. It really struggles. It really doesn't do it very well. So we've implemented a sort of AI-guided algorithm that uses sort of protein-protein docking to try and predict ternary complex formation. And on the benchmarking we've done, we've seen some really nice results, and we can predict these ternary complexes with quite a high degree of accuracy. So the example I'm showing here is 6HR2, which is a VHL PROTAC. It's a very kind of well-studied PROTAC example. In the middle is the predicted Domainex structure, and then I've just kind of overlaid the two at the end. And you can see it's not perfect, but there's definitely a good agreement with the ground truth here. And these types of approaches that we can look at predicting these ternary complexes can help us optimize the protein of interest, but also look at linker and warhead optimization as well. And yeah, I think just to kind of summarize, I think the computation and AI ML platform we've developed has been used successfully across a wide variety of areas in drug discovery, all the way from target assessment, target ID, all the way through to kind of late lead optimization. And we've applied it to a wide variety of targets and modalities now, and seen success in most of them. I won't say all of them, but most of them. And what we're hoping to do is continue pushing the boundaries and hopefully enable the discovery of novel therapeutics for these challenging targets, and really just to kind of expedite traditional design, make, test, analyze cycles to keep pushing that. So hopefully we make these discoveries quicker as well. That's my last slide, so thank you.